the London School of Hygiene and Tropical Medicine podcast. Thanks for downloading the September podcast. This month we'll get really disgusting.
You are going to remember for a very long time that a poo has been in contact with the biscuit.
We'll find out about a meningitis study in Chad, which has produced some very positive results.
I've been at this game for a long time, and this is probably the most dramatic results I've seen.
And learn how one of the best treatments for malaria may not be as effective as we thought.
The drugs rapidly push down the parasite population. In about a third of children, a few remain, and in a good proportion of those, they come back.
To start this month's Cleaner Than Clean podcast, here's some less than clean words. Feces, urine, toilets, sweat, spilt blood, vomit, dirty nails, rotting flesh. Why is it that these words have the effect to make you feel disgusted? Dr. Val Curtis is the director of the Hygiene Centre at the school and has written a book called Don't Look, Don't Touch, The Science of Revulsion. The focus of the book is the evolution of disgust and how these feelings of disgust can help improve public health. Kat Arney washed her hands with soap and discussed with Val.
Why do we feel disgust?
It's a very powerful emotion, and it's one that even me as a disgustologist, I've been working in this stuff for 20 years, it still affects me thinking about revolting, nasty things. It's an ancient emotion. It's not a rational or logical emotion. It's one that's there because our ancient ancestors who had a good sense of disgust, they kept away from all of those things. So if you just imagine for a minute that you had an ancient ancestor who said, "Oh, I love poo. Oh, give me poo. Give me a taste of it. Oh, glass of urine. Oh, yes, I love it. Oh, vomit. Oh, give me a bite. Oh, that person isn't going to be very successful in life. They're not going to be very good at attracting mates, and they're not going to be very good at bringing up children. And it's the bringing up children that matters. So somebody with a very high sense of disgust is going to be squeamish about things that have microbes in them. And therefore, they're going to have more children compared to people who go, "Oh, I like disgusting stuff." So your ancient ancestors all had a good, healthy sense of disgust. The ancient ancestors that didn't, their genes died out.
So it's basically something way, way back in our evolutionary history that's almost evolved to save us from parasites and infections.
Well, it's actually older than humans. If you go way back to animals, they've all had to deal with the problem of how to not be eaten up by parasites. In fact, parasite pressure is probably more important, evolutionary speaking, than predators. So, we're used to the idea that an animal and humans have fear, and that keeps you away, keeps you safe from big animals who might want to eat you up from outside. Well, disgust is parallel. It keeps you safe from those little microbes, viruses, parasites that climb inside you and get a lift, a hitchhike inside you, and actually eat up your nutrition and maybe make you sick.
I'm just going to bring in a little plastic poo here.
It's really nasty, isn't it?
So obviously you can show that to someone and they're going to look revoltedly at it. How can you actually scientifically measure disgust in people and what people do find disgusting?
There's multiple ways you can do it. So, for example, I could put you in an MRI scanner and then I could show you lots of things like this, and if it would work even more strongly if I could sort of waft nasty smells down the MRI scanner for you, and you would see certain parts of your brain lighting up. Other ways you can do it is simply by self-report. With my PhD student, Mikhail Demara, we've done a large number of internet-based studies offering people lists of things that are disgusting and asking them to rate how disgusting they are. And that's been very, very interesting. We started off with a hypothesis that probably disgust would be elicited by things that got into your nose, things that got into your mouth, things that got in through your skin, and things that got in through your genitourinary system, through sexual contact, for example. And it turned out that if you do studies that way, that in fact there's 7 different types of disgust, not exactly as we predicted, a little bit different. So we tend to find people who are unhygienic, very disgusting. We tend to find things that have come into contact with unhygienic things. So if I gave you a biscuit and then put this down on the biscuit, you are going to remember for a very long time that a poo has been in contact with the biscuit.
And how consistent is this around the world in terms of, the things people find disgusting? I should imagine poo is pretty consistently horrible. But in terms of some of the other things, do different cultures find different things gross?
Much of the previous work on disgust came to it from the perspective that that people in different cultures find different things disgusting. And so the assumption was that, in fact, disgust was shaped by our culture, not by our innate instinctual selves. Well, actually, when I first had my eureka moment working on disgust, I'd been looking at stuff we'd collected from India, from Africa, from different countries in Europe, and from a multinational survey in Athens International Airport, actually. And what struck me from all of those things was actually, though there was some local variation, actually it was the same.
So this passion for poo and disgust, how can you use the information that you're gathering to actually improve public health?
Well, I want you, for example, to wash your hands with soap. It can save millions of lives, literally around the world. And very few people are washing their hands with soap. So if I can come up with ways of making you feel that your hand maybe is disgusting, maybe you have come into contact with something nasty, that is a powerful way to encourage you to wash your hands with soap. Same with toilets. We're working on campaigns now to try and get people to get toilets, because 40% of people in the world still don't have them. I know, it's still defecating. A billion people are still defecating in the open. Now, poverty is part of the problem, but it's also just it's normal in many societies to do that. So what we're trying to do is make it not normal. So one of the ideas we have for a campaign is to show very graphically how when someone goes to the loo in the open, something coming out of it's this contamination concept with disgust. This contamination is spreading. And it's spreading out from the field, it's spreading on people's feet, it's spreading on people's clothes, it's spreading on people's hands, it's getting onto people's face. Yeah. Now you see, very suddenly, instead of being an invisible topic, which people don't talk about, becomes something shocking, something visible, something important, that's salient to people. And we hope that using those sorts of approaches will really make people want to talk about getting toilets and ultimately actually spend the money on getting their own toilets.
So the book, Don't Look, Don't Touch by Valerie Curtis is published by Oxford University Press and is coming out now, and there's an event publicising it.
The event's called Discover Disgust, and it's for anybody who's interested in the topic. It's on the 26th of September in the evening. You're invited to come and hear a bit about disgust and why it matters, but you'll also be invited to participate in a study looking at your own disgust responses. And if you're really lucky, you can participate in our lucky dip, which has got, in fact, we're calling it an unlucky dip, and there'll be items for you to play with and take home with you if you wish.
Is that going to involve plastic poo?
It might, yeah.
So that's 26th of September at the London School of Hygiene and Tropical Medicine.
Yeah.
Thanks very much, Val.
My pleasure.
The London School of Hygiene and Tropical Medicine podcast. That was Kat Arney with Dr. Val Curtis, whose book, Don't Look, Don't Touch, is available this month. And you can see some of Val's disgusting things in an extended video version of the interview by visiting us on the web at lshtm.ac.uk. For more than 100 years, the Sahelian and Sub-Saharan regions of Africa have been blighted by periodic, large and unpredictable epidemics of meningococcal meningitis. In Chad, for example, although the mortality rates for outbreaks slowly dropped from 75% in the 1930s to around 10% in the 1960s, there were still regular outbreaks affecting thousands of people in the first decade of the 21st century. In the past couple of years, a new vaccine, PSA-TT, has been deployed across the meningitis belt, and its effects have just been published in a paper authored by Sir Brian Greenwood, who told me more about the project and the results.
Only a small proportion of the people who get infected get meningitis, and in most people the bug just lives in the throat and what we call an asymptomatic carrier. And therefore, if we're going to study really what the impact of the vaccine does, we have to look at both the patients who get meningitis, but also the healthy people, to see whether they are carrying this bacteria in the throat. And this is what we did in the study in Chad.
And that was really the motivation for the study?
And that was the
was it?
motivation. So we started off this group, which has, I'd say, 7 partners from 7 countries in the meningitis belt of Africa, and then a group of colleagues in Europe, in the UK and elsewhere in Europe. And we set up this group about 5 years ago because we knew that we knew this vaccine was coming, and we knew that looking to see whether it would have an effect on carriage was a very important part of its evaluation. And it was at the right time the vaccine was introduced. The only country in which there was a lot of, or the only area in which there was a lot of serogroup A meningitis was in Chad and in the next door neighbouring bit of northern Cameroon. So we had a sort of unique opportunity there to see if the vaccine was stopping an epidemic.
It's a large area.
Mm-hmm.
Can you tell us a little bit about perhaps some of the challenges you might have
It
met?
is a very large area. It's a very large country. It has had a fairly rough time politically and socially. So there aren't very strong health system there. The majority of the people do live in the southern area, not up in the northern sort of more deserty parts, and that's where we sort of focused our studies. But there was an enormous willingness to be involved in this project to help. Doing a carriage study of 6,000 people is not a small undertaking, doing all the microbiology of that, and they all coped really well.
Presumably samples had to be sent
yes,
miles
they had to
away.
come 100 miles from the area where we did the carriage studies. So the sort of level of surveillance that we got over the whole of the country was not of the standard that you would ideally hope. But the logistics and the cost of doing that would have been impossible, and we didn't have enough money to do that. So we just focused on the areas where the vaccination had been done, which were about 2 million people near the capital. And even though the surveillance was much higher there than in the rest of the country, even so we found this dramatic effect, and meningitis almost disappeared. Whilst it was going on quite a lot, the epidemic was spreading in the rest of the country, and I'm sure if you'd had very good surveillance there would have been even more cases, and the effect would have been even more dramatic.
The vaccine was administered across Chad.
Yes.
Over how long? I mean, how long?
It was done about, I can't remember exactly, but sort of within a month or so. I mean, or perhaps even less than that, perhaps a couple of weeks. So this is done as a mass campaign, and just the people aged 1 to 29 were vaccinated. And what was really encouraging is there wasn't any meningitis in the older or the younger ones. So this is an example, as we were discussing, of what we call herd immunity, that if you can vaccinate a high enough proportion, then you can protect everybody because you stop the bug being there. It gets rid of the bug, and so even the people who've not actually been vaccinated are protected.
So the results, I think we've basically covered the results. It's a good thing.
It's very, very dramatic. So I've been at this game for a long time, and this is probably the most dramatic results I've seen from any intervention. Because it probably is nearly sort of 95% effective. So you give one shot of a relatively cheap vaccine, and you could have this impact on an epidemic. I mean, it is pretty dramatic.
So, are you surprised at those results? Or
Not entirely surprised. I'm very gratified, that's happened. Because we know these conjugate vaccines are very effective for that. And I mean, the other vaccine I worked in quite a lot involved with is the malaria vaccine. And that's a really difficult. We're struggling with 30% protection, 50% protection. Can we get it up a little bit? And I mean, that may well be worthwhile, but to then suddenly have a 95%, this is really very, very impressive.
So what happens next? The paper gets published.
I mean, the vaccine is gradually being rolled out across all the meningitis belt countries, and hopefully there will be money for that, because the money has come from, at least some of it's come from the international donors through Gavi. I think it seemed to be a pretty good investment, but bugs fight back. There are several different serogroups of the meningococcus which can cause meningitis, and we've only knocked out one, the most important one. So there is a worry that some of the others may come and sort of fill that gap and cause epidemic disease, and we know that they can do that. And therefore it's absolutely key that surveillance continues to see what happens.
Is your sort of hope that eventually there'll be one vaccine that can cover?
I think so, yes. That would be what one would hope. So I think this was the first. It's not the end of the story. No, it's a very important one, and sort of something like 75 or 80% of these big epidemics have been caused by this serogroup A. So I mean, if we can knock those out,
it's a huge first step.
It's a huge first step. But bugs fight back. One has to keep up the enthusiasm. We don't know how long the protection will last. I mean, it might last 10 years or something. In which case, then you would have to have some sort of catch-up campaigns. Would there be enough money to do that? And also there are still some uncertainties.
That was Sir Brian Greenwood. From one study with positive results to a malaria study which worryingly may have negative consequences. Artemisinin-based combination therapies, or ACTs, are now considered to be the best treatment for malaria. However, a new study in Kenya has suggested that ACTs did not clear all the parasites in some patients, and that these uncleared parasites can be readily passed on. Julian Mayers spoke to Dr. Colin Sutherland to find out more.
ACTs were deployed in some parts of Southeast Asia at the end of the 90s and into the early noughties. In Africa, ACTs were not really deployed widely until a few years later. But by 2006, 2007, they were adopted as first-line policy by most African countries. Two different groups working in Cambodia in 2008, 2009 noticed that malaria patients they were treating with IV artemisinin for severe malaria, they were clearing the parasites much slower than had been the case. And they were worried this was something new.
What was the limitations of the Cambodia study which led to the Kenya study?
So there were two main things. Firstly, in Cambodia and then in the follow-on studies in the countries around that area, they were looking at adult malaria cases, hospitalised and treated with intravenous artesunate. And the second thing is, to make it happen, they took a blood sample from each patient every 6 to 12 hours. So it's a very high frequency of blood sampling. Now in Africa, malaria is mainly a disease of children. So we reasoned that it would be very difficult to convince ethics committees that we should take very small children and take a blood sample every 6 hours by finger prick or venipuncture. And secondly, very few African malaria patients are hospitalised and get IV treatment. It's oral, and it's oral combinations. So we developed a new protocol for looking at combination therapy.
So how did you go about it?
Well, the reason that the studies in Southeast Asia took blood samples so frequently was that they were using microscopy, looking down a microscope at the blood film to count the parasites. We developed a DNA-based quantitative test. It's not dissimilar to the techniques police use in a crime scene. A very small trace of a biological sample can give a DNA signal that can be interpreted. So we could take far fewer samples and still get an indication of how quickly those parasites were disappearing after treatment. The clinical work in the field was led by Patrick Sawa from Mbita in Kenya and Dr. Tern Balsma, who's our colleague here at the London School. And Tern and Patrick enrolled 300 children and they randomised them to two different ACTs. So we had two treatment groups, but both received an ACT.
And how were those children monitored?
Well, we did the usual monitoring, which is a finger prick daily for the first few days, and then another one weekly out to about 6 weeks. And we were surprised to find that a large proportion of these children still had detectable parasites by this method 3 days after treatment. The microscopy was telling us there were no parasites. This looked as if we had no slow clearance at all, but when we brought in the DNA-based test, we suddenly found evidence of parasites below the level of detection by the microscope.
So it might not be a case that the drugs aren't working as you first thought, it's just that you weren't detecting them, the threshold was too low.
That's right, and you've put your finger on the interesting question. Is this something new, or has this always been the case with apparently effective drugs? Actually, they always leave this small residual population of parasites. One of the ways to partially answer that question is to see if it was roughly 35% of the patients, those that had this extra residual parasite population still there at day 3, did it mean anything? Did it impact on the 6-week follow-up? And the gist of this paper is to say that actually it's really important. And there's a very strong statistical correlation between having a detectable parasite at day 3 by this very sensitive technique and being infective to a mosquito at day 7, or positive by microscopy at day 28 or day 42. So the drugs rapidly push down the parasite population, and in about a third of children, a few remain, and in a good proportion of those, but not all, they come back.
That's quite worrying, isn't it?
It's worrying. In one way it's worrying, but in another way it may explain some of the things we haven't quite understood about the patterns of parasite recurrence after treatment. It may explain why when parasites are seen 6 weeks after treatment, and most of us would have considered that to be, they've had another mosquito bite, it's a new infection, we actually find that they are often show evidence of being selected by the drugs. They have a genetic signal of being different from the population as a whole. So this is starting to get into the realms now of resistance.
What comes out of this study? Where next?
Well, now that we've identified this persistent small population of parasites at day 3, we've started investigating what their genetic signals are. Are they different from the starting population? Have the drugs affected those parasites so that the ones that survive are particular types? And we have some genes of interest that we're investigating that have come out of previous studies, either in humans or in mouse models of malaria.
And what are the public health implications for this?
Well, from the data we have so far, we're not really alarmed, because by all the usual criteria for judging the efficacy of drugs, these drugs have worked well, and we need to emphasise that, and we have emphasised that in the papers. But it does raise a little flag saying, new drugs soon, please. It takes a long time to develop a new malaria drug, and we must keep that effort going.
That was Dr. Colin Sutherland. As always, you can find out more information about any of the items on this month's podcast and listen to extended interviews by visiting us on the web at lshtm.ac.uk. Next month, we'll have more of the latest research and news from the school. Thanks for listening.